And Oliver is another Harvard undergraduate who then went to Stanford for his graduate work. And then he came back and he was at the Whitehead for a while. Correct me if I do all this wrong, Oliver. And then he went to UMass Medical in 2007, and they've been lucky enough to have him since then. And Oliver has done some really ground. We talked about today. And so here you go, Oliver. Thanks.
Well, thank you so much for having me. It's lovely to see old friends and make lots of new friends. Yeah, let's get started. I've been interested for the entirety of my career in the process of epigenetic inheritance. I'm going to start out by defining my term here because the word epigenetic has been sort of polluted by the word epigenetic has sort of been used in multiple different ways from its from the sort of Waddington era, where basically everything that wasn't the genome was epigenetic. So like if it was hot outside and you were sweaty, that was epigenetic. It's sort of a useless term. I prefer the 1970s. Let's see, is this going to advance or like? No.
Oh, is it plugged in? Did somebody plug it in?
Okay, here it goes. Okay, so I like to use epigenetic in the 1970s Riggs and Holiday definition, which is that a trait is epigenetically heritable if two cells or organisms with the exact same DNA differ in some trait, and then they can pass those phenotypic differences on to their progeny without any difference of DNA sequence. And the cocktail party version of this is cell state inheritance, right? So even though all the cells in your body do first approximation have the same genome, when a liver cell divides, it never makes a skin cell or a kidney cell, even though they share the book of instructions, it makes two liver cells. So state liverness is epigenetic, okay? So I started my career working in yeast. I now work in mouse, which is what I'll tell you about. But I've always been sort of interested in the mechanics, basically the when, where, why, and how of epigenetic inheritance. Now, decades of okay. Decades of study of things like cell state inheritance have identified three major carriers of epigenetic information. Two of them are illustrated here. This would be DNA modifications, the fifth base, cytosine methylation, as well as the packaging of the genome into chromatin. This is where my sort of technical expertise is in from yeast. And so this shows up, for example, in the Drosophila screens for that identified polycomb and trithorax by Ed Lewis. Those turn out to be chromatin modifiers, and so that's the foundational evidence that chromatin plays a role in remembering what kind of cell you are. And actually, the one that's not on this slide is the one I'll be talking about the most, which are small RNA pathways, which we'll get to. Now, this is, of course, cell state inheritance is a pretty straightforward, relatively straightforward system to look at. But as multicellular organisms, this is not how we divide. So in order for epigenetic information to make it from one organismal generation to the next, that information has to survive or be generated during the disruptive process of gametogenesis. All of that information present in the two germ cells has to be sufficient at the spark of life, the moment of fertilization, to then sort of unravel over the course of development to make a physiologically coherent phenotype. Okay, and so this is a much more difficult process going through the germ cells than just dividing and sharing a lot of cytoplasm in the case of mitotic epigenetic inheritance. Now, there is good evidence for epigenetic transgenerational epigenetic inheritance, is what people call it. It was first discovered in the 1920s or 19-teens, I think, by Bateson and Pellou in their studies of the rogue garden pea. This is the 1950s update of this. Brinks and Co. in the 1950s named this phenomenon paramutation. And so the way paramutation works in maize, which is where it's been best studied, you have two kinds of corn, purple and green. When you mate a purple corn with a green, so these are, when you mate a purple corn with a green corn, you get green offspring, okay? Now, if this were a Mendelian genetic trait, this would be big B, this would be little b. These green progeny would be heterozygotes, big B, little b. So what happens if you back cross this to little b in this scenario, you should get half purple offspring and half green offspring. And what you get is entirely green offspring. And this holds true through hundreds of generations of back crossing. So the reason they called it paramutation is that they had this idea that one piece of DNA affecting the other piece of DNA. And so this is sort of the original epigenetic inheritance model, worked by largely Vicki Chandler over the second half of last century, have sort of done a lot of genetic screens to identify the molecular basis for this epigenetic inheritance. And it turns out that all three of the epigenetic information carriers I told you about, DNA modification, chromatin, and small RNA pathways, are all responsible or required for this multi-generational silencing. And they believe, for a variety of reasons, that the thing that's transmitted in the germ cells is probably the small RNAs that arise from these repeats. Okay, so the fact that some epigenetic information, although most of it doesn't, but the fact that some epigenetic information makes it through the germ cells into the next generation sort of led to a lot of chatter about resurrecting an idea that's a long discredited idea, often called Lamarckian inheritance, or the inheritance of acquired characters. And the sort of brief idea there is that there are some environmental conditions under which it makes sense for parents to tell their kids the world that they experienced. And because, unlike DNA sequence, epigenetic marks are typically environmentally responsive, right? So if you heat shock yeast, the chromatin architecture of the genome changes completely. Small RNA levels are affected by various perturbations, same for DNA methylation. So because the epigenetic information carriers listen to the world and sometimes make it into the next generation, this sort of resurrected the possibility that the environment you see in one generation could modulate traits in future generations. So, like I said, this is often called Lamarckian inheritance, although that's a misnomer for multiple reasons. It was sort of the beginning of the end for this idea in polite society. Came from August Weismann. He was a German embryologist at the end of the 19th century. He defined the soma germline distinction, right? The eternal germline that stretches back billions of years. The soma is the meat machine that carries germlines around trying to find other germlines. So what he did is he cut the tails off of mice, mated them to each other, and measured tail length in offspring. And he sees no change in tail length in the kids. And so this was a very influential refutation of the inheritance of acquired characters. Now, looking back, we can think that there's probably a problem with this experiment, right? Maybe the useful thing to do if your parents tell you that someone's going around cutting tails off isn't to have a shorter tail, it's to run away from people in lab coats. Even in the environmental milieu of the time, this is a little bit of a rigged experiment. Lamarck, like anyone who thinks about this for more than 2 minutes, recognized that soldiers from wars throughout history who've lost a limb don't have children with flipper hands or something, right? People still have their arms. Lamarck dealt with this theoretically by arguing that you would only tell your kids about things that were useful, or that you wanted to tell your kids about. What wanting means in this context, we don't know. So anyway, mutilation is probably a little bit of an unfair experiment to be testing this hypothesis on. Okay, so that's the end of the sort of historical aspect of this. I bring this up still because basically what we and a bunch of other people have done over the last 20 years is revisit this form of experiment, poke an animal and look for something in its kids, but with different perturbations and with different measurements. Okay, so we do this all as paternal effects. Of course, in mammals, mom is baby's first environment. So, fetal alcohol syndrome is not magic and epigenetic, you're just poisoning the fetus. On the other hand, so it's very difficult to disentangle, molecular contents of oocytes from the fetal gestational environment. You can using IVF, which we're not doing, but certainly before we were reproductive biologists, this was a daunting idea for us. But with dads, you can just, set up the mating so dad leaves sperm in a membrane. And so we do this as paternal effects. What we do is we split male litter mates to, we've now done quite a few environmental conditions, but our first one was control and low protein diet. We leave them in the cage with the female for 1 to 2 days. So they're certainly not directly interacting with their offspring. So there's no meme or cultural inheritance. And we're trying to minimize the extent to which they transfer, say, microbiota through defecation in the cage. We let the females be pregnant, have kids. At 3 weeks of age, we grind up the kids, and we measure gene expression in the livers, actually. And so the two big differences between our experiment and the classic Weissman experiment, one is that this environment is chosen so it would be plausibly useful to tell your kids about, and I can get into that in questions as to why we would say this. And the other is, instead of measuring one quantitative character in kids, we're measuring 30,000. So what we find is, this is typical microarray heat map. This is back in the microarray era before deep sequencing. Every column is a comparison of a low protein offspring and a control offspring. What I mean is low protein dad, controlled dad. The way we treat the kids is completely identical. And the low protein RNAs were labeled in red and the control in green. So basically, these are all the genes that are upregulated in kids, and these are all the genes that are downregulated in kids, depending on what dad ate. And as you can see, this is highly penetrant, but not completely penetrant. When you look at the kinds of genes that are changing in kids, these are nothing in particular, although there are some circadian genes, I think period 2. These are very heavily enriched in cholesterol and lipid biosynthesis, okay? And to see whether or not this is a physiological phenotype versus just a gene regulation phenotype, we carried out metabolomics in these livers. What we see here is that the low protein offspring have significant decreases in free cholesterol and cholesterol esters. This is as far as I'm going to go in terms of phenotyping these kids. But for people who know about metabolism, this suggests a bile acid phenotype. And so potentially, and we haven't looked into this at all, maybe what the kids are trying to do is obtain more nutrients from their food by dumping more bile acid, and they're homeostatically compensating by upregulating cholesterol biosynthesis to keep up with the loss of cholesterol. So maybe that's the phenotype. We really are not physiologists, we're molecular biologists. So this is not what we're going to get into too much. So we published this paper 13 years ago, and it was sort of a scary moment for me in my career because I had a perfectly functional career in doing chromatin work in yeast. Over the next, the last 13 years, there have now been something like 1,000 papers documenting paternal effects of diets, of stressors, and toxins. Okay, so I point these papers out. So this is a paper actually that pushed us out the door. This was high fat in rats affecting glucose control in kids. This is basically the same system as ours a couple of years later, showing that also in addition to glucose control, there are things like blood pressure and one I already lost the phenotype, but there are other cardiovascular phenotypes that respond to paternal diet. These are all very interesting papers to me because what these guys did is they starved a pregnant mom, and then they used her son as the paternal generation. And so these people all sort of knew more about epigenetics. Well, I knew this, but they chose more wisely than we did. So imprints, the cytosine methylation that's male gamete or female gamete specific, are established in primordial germ cells. That occurs during fetal development. And so these systems, they were trying to actually more directly impact the establishment of imprints, whereas our perturbations and all of the postnatal perturbations are messing with germ cells after imprinting has been established. Okay, And I think there are really interesting differences between these kinds of paradigms. Just to sort of snow you with titles, these are some of the papers about stress. And then the third great arm of paternal epigenetics are various toxins, whether taken by choice, like cocaine and nicotine, or things like endocrine disruptors like vinclozolin, which actually Michael Skinner's was the first paper in this domain in 2005. And actually the last one I don't mention here, but also advanced paternal age is correlated with autism and schizophrenia in the next generation. People often think about that in the context of mutation, but I think it's just as likely that it's going to be an epigenetic. So these are all rodent studies. There is some evidence for this kind of stuff in humans. The famous one that everyone talks about is the so-called Dutch hunger winter. This was at the end of World War II in response to a transit workers' strike. The Nazis starved the Dutch people, burned the fields and salted the earth, burned the warehouses, salted the earth. Government rations were down to 700 calories a day. People were eating tulip bulbs to survive. Something like 20,000 people starved to death. So, and this persisted until the Allies restored food control in like March of 1945. So what's unusual about this is it's rare for a first world country to go into and come out of a famine with defined kinetics. And what people have done over the decades since is measure a bunch of outcomes in the next generation. And what they find is significantly elevated rates of diabetes, obesity, and cardiovascular disease in the children who were in utero during the Dutch hunger winter. Okay, so this has led to something that's called the thrifty hypothesis, which is that if your parents tell you you're going to go hungry, it makes sense to hoard every calorie you can get your hands on. When conditions of plenty return, this can be maladaptive. Okay, so now, of course, this is a direct effect on fetal growth, right? These children were in utero during the starvation, which, as I said, is difficult to disentangle epigenetic from direct action in this case. There's a little bit of evidence in humans for a male line effect from the so-called Uvercalyx cohort. This is where a remote parish in Sweden where they started keeping really good crop records in the 19th century for tax purposes. And what Bygren, Kati, Pembry, and others have done is track modern morbidity and mortality to ancestral food access. And what they find is that your paternal grandfather's access to food affects diabetes, et cetera, in his grandson. So paternal grandfather, male line ×2. So there's a little bit of evidence for this in humans. So that's all of the phenomenology I'll tell you about. So this sort of recaps what we're interested in as molecular biologists. You can put males into a variety of different conditions, mate them to control females, and measure altered outcomes in the kids. Now, the natural hypothesis is that this is going to act through one of the three epigenomes, chromatin, DNA modification, or small RNAs. It's worth pointing out there are other ways for dads to talk to kids. Seminal fluid can affect all sorts of female post-copulatory molecular biology and behavior. This is very well established in Drosophila from Mariana Wolfner's work. Some evidence in mammals for this. Moms can judge mate quality and change how they allocate resources to the kids. This is a great literature in birds, particularly the Goldian finch, for reasons that we can get into. So it's, in principle, moms could be judging our low-protein dads and restricting blood flow to the placenta. And finally, I don't have it on the slide, but the microbiome is another way that dads can affect metabolism of their kids without it being sperm epigenetics. So how do we distinguish these? Of course, the simplest way to do it is assisted reproduction. So we take sperm from control or low-protein animals, fertilize and control oocyte in vitro, and then transfer it to a second female. And we've been able to recapitulate our cholesterol phenotypes through IVF, which tells you that definitively that the information is in sperm or co-purifies with sperm. And actually, pretty much all reproduction in our lab now goes through IVF, which is a bummer for the mice because that's all they had. But it also allows us to take oocyte epigenetics seriously now because we are now splitting moms into the oocyte donor and the uterus donor. And so we've started to get a little bit more into the egg as well as sperm. So the title of my talk was Epigenetic Contributions of Sperm to the Next Generation. I'm going to just do one slide each on problems with chromatin and cytosine methylation, and then I'll tell you where we are as far as small RNA as a possible explanation here. Okay, so in terms of cytosine methylation, of course, we know that cytosine methylation can be transmitted from parents to kids in the case of imprinted genes, right? So these are genes that they're either only expressed from dad's copy or only expressed from mom's copy, which means that they remember which germline they came through. Now, the problem with cytosine methylation in an environmentally controlled system like we're talking about is Dr. Keefe showed these images of cytosine methylation reprogramming, which occurs in primordial germ cells and at fertilization. So virtually everything is erased at fertilization, except for the imprinting control regions. And they're actually hard-coded by a sequence-specific DNA binding protein called ZFP57. So in other words, if you don't have a ZFP57 site around, no matter what happens to the sperm methylome, it probably is getting erased. So that's one major problem with cytosine methylation in sperm as a carrier of environmental information. Another problem with this is that all of the studies that have sort of claimed cytosine methylation changes that they think have something to do with the next generation tend to see like a 10% difference in methylation. And so that could have physiological consequences in a multicellular tissue like the liver, right? If you go from 10% to 20% methylated, or say 80% to 90% methylated, you can have the number of cells expressing growth hormone, and that can matter for the organism. But sperm are not a multicellular tissue. Every sperm acts alone in the end, except in like deer mice, but still only one genome fertilizes the egg. So for these 10% methylation changes to affect kids, you would have to imagine that it would only affect the penetrance by a small amount within the litter of some phenotype. Okay, so these are two of the big problems with cytosine methylation. I'm not saying it can't be the answer to paternal effects. I'm saying it has a lot of issues to overcome. Chromatin has been claimed to affect offspring traits. So there are two fairly high-profile studies where people either overexpress or delete histone-modifying enzymes in the testis in the developing germline. And they see all sorts of phenotypes in the kids, skeletal abnormalities and increased cancer risk and stuff. The problem with chromatin in sperm is that basically sperm eliminate almost all of their histones. They retain, depending on the mammal, 2 to 10% of the nucleosomes. They're largely packaged by these little peptides called protamines, which make a very compact sperm head. And so there's been a lot of interest in where the nucleosomes are retained. People speculate that where nucleosomes are retained programs early embryonic gene expression. This is sort of a review from Victor Corses showing the old view and the new view based on his work. The big problem with this whole field is that everything that people have measured about sperm chromatin turns out to be wrong, at least in mouse. It turns out that what happens, and this is what we're trying to publish now, when you take sperm out of the epididymis, it's polluted with cell-free chromatin. It's 2 to 5% contamination. But because sperm only have 2% nucleosomes and you have 5% contamination by a fully nucleosomal genome, that's actually what people have been measuring. And you can very easily clarify this by treating sperm with DNase before you break them open. And it completely changes the profile of chromatin you see. And I just like this. Azusa Inui made this charming cartoon about what we're talking about here. So people are looking at sperm. We don't know what's in sperm, and people are thinking that this cloud of contaminating material is the way things are organized. Okay, so I'm going to spend the rest of my talk on small RNAs, which is what we have focused on. Small RNAs also have a bunch of problems. So I'm not going to say that I think small RNAs are the answer to paternal effects, but the field certainly, by and large, has been going in that direction. Now, the big problem with sperm RNAs, you can immediately see here, is that sperm deliver almost nothing in terms of molecular contents to the zygote, right? The oocyte's 1,000-fold larger at least. And of course, I also think, although I'm not sure of this, that sperm contain fewer RNA molecules on a per femtoliter basis. So they're really just delivering a whisper of RNA to the embryo. And this is some recent data of ours. We've gotten to the point where we can clone small RNAs out of pre-implantation embryos. This is just for microRNAs, a scatter plot of oocytes versus zygotes. You really don't see a big change once you become a zygote. So once you've received a sperm load of stuff, okay? So that's the big problem with sperm RNA. That's one of the big problems. I'll touch on a couple others as well. The reason people have focused on small RNAs, one is that they are the transgenerational epigenetic information carriers in the really good systems for it. So paramutation in plants, RNAi in worms, which of course is near and dear to UMass's heart. There's a sort of a reason based on history in other organisms, although those organisms are different from mammals in important ways. But also RNAs are the easiest to test functionally, right? So a number of studies, including ours, which I'll tell you about ours, but these are a bunch of other people, will sort of make control zygotes by ICSI or IVF, and then they'll take either purified or synthetic small RNAs. So they'll take control versus high-fat sperm, isolate the small RNAs, and then microinject them into paired control zygotes. And they report being able to recapitulate many of the phenotypes they see in the natural mating versions of these paradigms. Okay, so now I don't believe many of these studies. A big problem with a lot of them is that they're almost certainly injecting a lot more RNA than sperm naturally deliver. But at the very least, they tell you that RNAs that sperm have, in principle, could affect the next generation. So this is the reason I think small RNAs, but it's just a convenience reason, right? It's just a lot harder to do the same kind of experiment where you write cytosine methylation onto base pair 57, right? So to some extent, I think this is people are just looking where they have the ability to look. And I say that as one of the people doing this, but I think it's just worth pointing out. Okay, so let's talk about small RNAs in sperm. So sperm carry a very unusual payload of small RNAs. So this is when you clone RNAs less than 40 nucleotides, this is the distribution of sizes of the stuff you clone. This peak at 21 nucleotides, these are microRNAs. These would be basically everything you would get out of sequencing small RNAs from a hepatocyte, a fibroblast, or something else. Sperm have about 10% of their small RNAs are microRNAs. The elephant in the room is this giant peak from 28 to 35 nucleotides. All of these are made from the 5' halves of tRNAs, although, as I'll get to in a little bit, we're missing the 3' halves. It's not that they're not there. We can now see them. But for a long time, the only thing anyone saw were basically these 3 5' tRNA fragments with TERFs. Okay, so they're clipped in the anticodon, and the 3 most abundant are glue-CTC, Val-Cac, and Gly-GCC. And so what we see when we compare control and low-protein sperm is about a 3-fold upregulation of TERF Gly-GCC. Okay, and so this is why we got in. This is why we chased small RNAs in sperm. There are a bunch of reasons here. First, like I said, sperm deliver very little RNA to the oocyte. So in order for something to be meaningful, it's really going to have to be one of the most abundant RNAs in sperm. This is the 3rd most abundant RNA in sperm. 2, small RNAs are the way parents talk to kids, and tRNAs very directly see metabolism. So of course, you would tell your kids about diet by a tRNA piece. And the last one, and this one sort of gets at the theme of the first 2 talks, is that tRNAs, and this also like cleanly separates people who are older or younger than 40 years old. tRNAs are universally used in LTR element replication, right? So this is a picture of HIV, but this is also true for endogenous retroelements, LTR retroelements. So the HIV RNA genome is packaged in the virus head with a tRNA bound to something called the primer binding sequence. tRNAs are the primers for reverse transcriptase in the wild. So you make, after infection, make cDNA to one long terminal repeat, come over to the other, make cDNA to the whole viral genome. This is so universally conserved among LTR elements, which generally speaking are going through a host pathogen arms race and rapidly evolving, that there's a sort of a very compelling hypothesis that the function of tRNAs in the RNA world was as replication factors, and they've been moonlighting in translation for 1 billion years. So the reason this interests us is that you can imagine that by clipping your tRNAs, you could fight off an LTR element, right? So either you could prevent a tRNA from priming reverse transcriptase, or you could load the pieces of the tRNA into an Argonaut and go clip the viral genome and sequences of homology. And so epigenetics, so people talk about piRNAs as a genomic adaptive immune system. Once you've been infected by a retroelement, piRNAs will fight off its spread. tRNA fragments could act as a genomic innate immune system. Any LTR element I could get from anyone in this room is going to have to use human tRNAs in its replication. By clipping my own tRNAs, I can fight it off. Now, epigenetics, generally speaking, is about control of selfish elements, first and foremost, which then gets co-opted for other forms of genomic regulation, like in the case of organ. So this is really why we got into the tRNA fragment business. Okay. Now, here. And so I'll tell you a little bit about the biogenesis of tRNA fragments and then a bit about the function. So the dogma about RNAs in sperm is that they're the degraded remnants of the RNAs you use to make a sperm. Because if sperm are just a swimming haploid genome, they don't need to do transcription or translation. And so you just like chew everything up. And that's a great way to prevent other things from infecting you if you're just destroying all of the RNAs of molecular biology. So the sort of null hypothesis here is that low protein testes have higher levels of tRNA glycine GCC. So we set out to test this hypothesis, and in the process of doing so, realized that tRNA fragments don't seem to be made in spermatogenesis in the testis at all. Okay. So I don't need to introduce this audience to epididymis, apparently. So as you all know, after sperm leave the testis as a morphologically mature spermatozoa, they still don't know how to swim yet. And they spend, depending on the mammal, a week to 2 weeks going through a 1 to 10 meter long tube called the epididymis. From caput to corpus decata, this is where you get sperm for IVF. You can make these guys swim. And this is the storage organ for ejaculation. So muscle contraction squeezes out the bells. So this is a picture of the first description of the epididymis by Reinier de Graaf in the 1700s in Leiden. My favorite, so the epididymis is the least studied organ in the body. Doesn't get cancer, so it doesn't show up in any of the cancer things, which is sort of an interesting, I don't know, nobody knows why it doesn't get cancer. It's quite interesting. My favorite factlet about the epididymis is that in a 2004 German newspaper poll, it came in third among people's favorite male reproductive organs. Which is like, okay, first of all, we beat the prostate. Second of all, why would you do a poll like this? Like, what are the policy implications? Anyway, so this is an image we've taken of the mouse epididymis. I think it's very beautiful. So this is, the piece of tissue is a nugget about that big. And you can see how you get about a meter of tube all sort of squiggled up in here. There are a bunch of different septated compartments. There are 10 segments defined in the mouse epididymis. They have massively different gene expression in the secretory epithelial cells across this. And so what you really should be thinking about is the sperm are going through a series of 6 to 8 completely different luminal environments during the process of maturation. Okay. And we really have very little idea of what the purpose of all this is. They learn how to swim, but most of what people describe about the role of the epididymis are things like calcium signaling and pH balance and stuff like that. Ion signaling is something that takes milliseconds, and sperm are doing this for 2 weeks. So it's sort of unclear why sperm have to spend so much time going through this process. One thing we think is happening here is that we believe that this is actually the source of the RNAs that sperm are going to deliver to baby. Okay. And so this would be an example that shows up all over the place in reproduction of soma to germline RNA communication. The soma is often a place where dangerous molecular events take place, so that sperm can learn about stuff. Okay, so here's a little bit of evidence for that. So if you clone small RNAs from the testis or from any purified sperm population from the testis, they're filled with piRNAs, the genomic adaptive immune system. This is the data I showed you earlier. So sperm are filled with red. Here is the tRNA fragments. And really the last stage of sperm in the testis are still piRNAs. The first stage of sperm in the epididymis are filled with tRNA fragments. So this is a massive epigenetic remodeling event. We believe, this is not universally believed by everyone, but we think that the epididymis is synthesizing the RNAs that show up in sperm. So here are northern blots for tRNAs. You can see they're not cleaved in the testis. They are cleaved in the epididymal epithelium. We know that the epididymis ships little vesicles called exosomes in other contexts, but epididymosomes here. We know from people like Pablo Visconti that epididymosomes deliver proteins to sperm, but exosomes are generally known as RNA carriers. So we can isolate these and clone RNAs out of them. We call this epidemiological epididymosomal epigenomics. At some point, I'm going to have to do some epifluorescence. So this is a scatter plot of small RNAs in sperm versus epididymosomes. You can see that they basically have exactly the RNAs that sperm are going to gain. And the exception that proves the rule is that piRNAs, which sperm have carried forward from the testis, are below the diagonal, but everything else is really on the diagonal. And finally, we can recapitulate this delivery process ex vivo. So we can take testicular sperm, fuse them with capid epididymosomes, wash the vesicles off as well as we know how. Here you see the piRNAs have not changed, and everything above is an RNA that's been delivered to the sperm. Okay, this gets maybe a little bit more complicated than this talk needs to be, but I think at this point, so our hypothesis at this point, 5 or 6 years ago, was that the epididymis is where RNAs are being made to give to sperm to tell the baby about the world. I think it's probably a lot more complicated than that, in the sense that there's almost certainly RNAs that are carried forward from the testis, like piRNAs, RNAs that are made in situ in the sperm, as well as traffic RNAs. And we're trying to sort of sort through that mess now. Here's an example, and this is going to come up again. So here's the major epigenetic transition I told you about. So in the testis, you're looking at all piRNAs. Those get almost completely destroyed as you enter the epididymis. Most of the tRNA fragments are delivered very early in the epididymis. A handful of them are produced later in the cauda. So like Valcat, the valine tRNAs seem to be distal specific. So we sort of imagine that the different sectors of the epididymis are synthesizing different suites of RNAs to deliver to baby. For microRNAs, the interesting thing is that most microRNAs don't change abundance a ton over the epididymal transit process, but there's a really interesting set. All of them come in genomic clusters. They're often rapidly evolving, that are present in testicular sperm, present in cauda sperm, but completely absent in capid sperm. And these are really weird. There's something going on here. And again, this is something I'll be telling you about. But just to show you the data behind these things, I already showed you these pie charts. I've just added testicular. No, I had that too. So you've already seen these. Here's our favorite example of those clustered microRNAs. This is on a log scale. So here's the level in testis, here's the level in cauda, here's the level in capid. So they are at least 100-fold less abundant in capid sperm than they were in the testis or in the cauda. Okay, so these are really odd. And this particular cluster is particularly unusual. It's from platypus through humans. There's always a cluster of microRNAs here on the X chromosome between Fragile X mental retardation and slit RK2 or something. The seed sequences, which is how microRNAs work, completely scrambled, even from like mouse to hamster or squirrel or something. So they're really rapidly evolving. They seem to be derived from retroelements. They are believed to be imprinted. And for some reason, they're in the testis, they're gone in the caput, and back in the cauda. The sort of controversy at this point, and the other reason that I'm kind of weirded out about this hypothesis, if you dump these RNAs and then regain them in the epididymis, some of the RNAs that do this include like miR34C, like a very famous testis-specific microRNA. And it's sort of perverse for us to propose that a famous testis-specific microRNA in an ejaculated sperm was destroyed in the epididymis and then came back. right? So this is a problem with our biogenesis hypothesis, I think. The other problem is, if you think about how many small RNAs fit in an exosome, for the number of RNAs that we think are being gained, you'd have to at least double the membrane area of sperm during a time frame when they are, if anything, sort of winnowing down and becoming more hydrodynamic. So for these reasons, I'm a little bit skeptical of our own hypothesis for these guys. So this is the hypothesis I told you. You sort of make these things in the testis, then you degrade them or dump them in the residual body in the caput, and then you regain them in the epididymis. Two other possibilities here. One would be that you make a mixture of cluster-mere positive and cluster-mere negative sperm in the testis. And sperm, of course, in the cauda have to have gone through the caput, but that doesn't mean that most sperm in the caput are on their way somewhere. So it could be that the caput is pulling these sperm out of circulation. Now, I know that for the epididymis community, this is anti-dogmatic because they don't think there's any role for the epididymis in quality control. But in any case, that's a possibility for these dynamics. And then we had started thinking, and then Wei Yan published this, that maybe what was happening is that these RNAs are present in the cytoplasmic droplet, which all of you guys will know about. And perhaps what's going on is that the cytoplasmic droplet is easier to knock off of caput sperm when we purify them, and harder to knock off, or the microRNAs have now relocalized through the sperm in the cauda. Now, what I just said again is sort of the opposite of what people know. It's actually harder to, easier to knock the residual body, sorry, the cytoplasmic droplet off of cauda sperm. Anyway, these are sort of three different ways these small RNAs could go, and I just think it's worth acknowledging and highlighting when there's controversy in the field. Okay, so the second thing, and this I can see I'm sort of blithering a bit past where it should be, I'll just point out really quickly that we also know that the view that we, and I will say everybody who's ever cloned small RNAs out of sperm, what we have seen in sperm in terms of their RNA payload, we've known for a long time it's missing part of the picture. So we know that tRNAs are generally cleaved by an RNase A or T family member. This will leave a cyclic 2', 3' phosphate at the 3' end. This will not be clonable by typical ligation-based methods. You can resolve this by treating the PNK without ATP. And when you do this, you see slightly longer cleavage products. So these are probably most of what you're getting in sperm. The other big question has been what's going on with the 3' halves. Are they getting destroyed, or are they just not clonable? And by northern blot, we and Qi Chen has also seen, we can see the 3' halves. So we know by northern blot that they exist, but they're not being sequenced. Over the last couple of years, we've been collaborating with Kathy Pollins in Berkeley, who has a really interesting new cloning method called ordered 2 template relay. It's a complicated method, but it turns out it does, for reasons we don't really know, it does great at cloning tRNAs. So tRNAs are hard to clone, I should point this out, because they're just covered with nucleotide modifications, some of which block reverse transcriptase. In fact, that's part of how the retroelement replication works, is by this hard stop. For whatever reason, this reverse transcriptase doesn't have any trouble with these nucleotide modifications. We can prove in budding yeast that we can now clone intact tRNAs and and so now here are typical protocols for RNA cloning. These are the 5' halves of glycine GCC. When we use otter, we can now see the 3' half as well. And if we look at the spectrum of RNAs you see, this is what you get with Illumina TruSeq, and this is what you get with otter. So we go from 3 privileged 5' halves to basically all tRNAs, both sides. And so that also probably changes a little bit how you think about biogenesis. A little bit more. Okay, and so that sort of raises function questions here. We've been thinking about the functions of 5' tRNA fragments in isolation, but now that we know that the 3' fragment is there, does it have its own function? Does it inhibit the 5' function? Do they exist in a nicked tRNA complex and have an independent function? And these are all things that I think the field now has to sort of come back to and think about again. So I think I have something like 10 minutes here. So I'll tell you as much as I can about how much? 5. Okay, so we have four different ways of probing the functions of small RNAs in the pre-implantation embryo. One is to either purify or synthesize small RNAs and directly microinject them. Two would be to genetically eliminate certain classes of RNAs by eliminating biogenesis machinery. My ex-postdoc Colin has actually done some beautiful work with this recently. Three, and this was the one I thought was most elegant, but I think I won't talk about it just because of the time here, is that you can use the natural small RNA dynamics as a way to probe the functions of large classes of RNAs, right? So if you do ICSI with a testicular sperm or a cauda sperm, you're asking what a piRNA payload versus a tRNA fragment payload does. And this is, I think, the coolest version of this. And then the last is to use parthenotes as a blank slate because they don't have any sperm delivered anything. So it's a sort of sperm minus embryo. I'm always told not to call them embryos.
Right.
Okay, so this is the one I won't tell you about. What I will tell you is that for all of these things, what we then do is we do single embryo RNA-seq. This was our first 200 or something embryos. It's really nice here because when you do principal component analysis of this entire data set, you can't always interpret principal components when you do PCA. This one is very interpretable. So it nicely turns early development into a circle. It turns time into angle. And basically what you're looking at is from left to right. That's the degradation of the oocyte transcripts. So that's the elimination of the maternal load. And then here, what's happening is that these embryos are before zygotic genome activation. They turn on the two-cell program, the zygotic genome activation program. Those transcripts are high. Sorry, it shouldn't be negative, whatever. And then those transcripts are degraded. So these two principal components actually tell you very specific biological processes. So I'll skip the caput versus cauda. It's published in 2018. It's been subject to a lot of pushback because we couldn't make offspring with caput sperm. Several other groups can. It turns out that this distinction comes down to whether you make your sperm heads by sonication or by passing them through a fine needle. So everyone uses sonication. They can make caput-derived embryos. We used a fine needle. We thought it'd be less disruptive. We cannot make caput-derived embryos. So for those of you who have been caring enough to follow the work, that's I think the answer to this. Now, what the difference is between the shearing and the sonication remains an open question. But the way we do things, this is a robust enough gene expression program when you use caput sperm that we can actually screen for the function for the RNAs who are responsible for repressing this program. And so epididymisomal total small RNAs will repress the caput state. And then we can, by size selection, split microRNAs and TERFs. And you can see here that microRNAs do not, the TERFs do nothing in this context, but microRNAs are sufficient to reverse the gene expression problems with caput-derived sperm. And it turns out that it's this mere 880 cluster actually. Okay, the thing that I wanted to sort of end on, I think, or that I think people will be interested in this audience is the Parthenote thing. So we put together a huge study with something like 1,000 individual embryo data sets where we compared natural mating embryos, IVF embryos, ICSI embryos, and parthenotes, and carried out single embryo gene expression. What I'm going to tell you about are the parthenotes. So here we're just activating oocytes with strontium chloride and cytoplasm B to keep them diploid. So of course there are like 5,000 genes that are misregulated. We know that there are going to be a lot of problems because all the imprinted genes are misregulated. So here you can see all the natural mating, IVF, and ICSI embryos are all expressing these maternally silenced genes. So the paternal genome will express this, but the maternal genome will not. And in parthenotes, they have two maternal genomes, so they don't express any of these genes. There are about 100 of these genes, but all of them will then have lots of secondary effects. So for example, exist is imprinted, and so you don't do imprinted exit activation, and all of the genes on the X are overexpressed. So you go from one imprinted gene to hundreds of downstream targets. Now, the cool thing about parthenotes is we can then ask what happens if you inject sperm RNAs into them. And so the targets of our cluster or our favorite tRNA fragment are repressed if we inject sperm RNAs into parthenotes, okay, as we would have predicted. And more broadly, this is an amazing scatter plot to me. We're basically looking at we're comparing the effects of sperm RNA delivery by fertilization. So this is parthenote over fertilized versus sperm RNA delivery by needle. And you can see that the correlation here is 0.3 something, which means that you can explain 12% of the gene misregulation in a parthenote by the absence of sperm RNA. Okay, so I think this is very good evidence that sperm RNAs do have regulatory functions in the early embryo. Okay, finally, our favorite tRNA fragment, since I probably used those 5 minutes now, I'll just tell you that what it does is it turns out that it represses, and here's in an ES cell model, this also is true in embryos, it represses all the genes of the 2-cell MERV-L program. Okay, so this is the zygotic genome activation program. These all use pieces of MERV-L, we've heard about this already, as its regulatory elements. And as I told you, we got into tRNA fragments because of a connection to LTR elements. And now we have a tRNA fragment regulating LTR-driven gene expression. So this was very exciting, and it's a total red herring. The connection here has nothing to do with the sequence homology between MERV-L and tRNA glycine. First of all, the L in MERV-L stands for I use a leucine tRNA as my primer. Second of all, lots of these elements are solo LTRs, where the body of the ERV has been recombined out, so they don't have the primer binding sequence. Third, our tRNA fragment is a 5' fragment, and the homology for reverse transcription is at the 3' side. So how does it work? This will be the last thing I say. In general, ERVs are opportunistic about underassembled chromatin. Zygotic genome activation occurs when you run out of histones to repress the genome. And so what my postdoc Anna Boscovich discovered is that our favorite tRNA fragment supports histone production. It does this by supporting the output of Cajal bodies. One of those is the U7 non-coding RNA. And so by supporting U7 production, you support histone production and thereby repress the MERV-L targets. And so this is our working model, is that by delivering a small RNA that supports histone production, sperm are either accelerating or decelerating zygotic genome activation. And by either extending the amplitude or the perdurance of the totipotent state, we think that this may affect the cell fate allocation into perfected ERV versus inner cell mass, and therefore resource acquisition by placentation versus embryo birth per se. And I guess I'll skip this point. Here's the cartoon slide that Anne probably want me to leave on. And then here's the lab, and thanks for your time.
I don't see very many working models that are raw. We have time for a couple of questions.
There's a very prominent epididymal fat pad, which I noticed is not in your diagram, and I wonder whether exosome transfer from the fat pad might be important, especially if there are nutritional, maternal epigenetic effects.
Yeah, so I have somebody, I have a postdoc who came from a metabolism lab, and he's really interested in trying to figure out that question. It's kind of hard to know how to address epididymal fat versus other visceral fat. What we do know, so that image of the epididymis I showed, half of it was red. So that's a reporter animal that has a Cree-driven tomato, GFP, whatever. And this is a study we published this year. If you express Cree in the brain, either by AAV transduction or by double transgenic, that Cree, and this also Corrado works in this domain, that Cree will actually recombine in the epididymis. So the epididymis is receiving Cree RNA or protein. We think it's exosomal RNA, but we don't know. So Cree RNA made in the brain ends up in the epididymis. The other Cree drivers that do that, or the other Cree driver we found that does this is adipo-Q, so adiponectin. So it does seem that at least pre-RNA and possibly other RNAs are making their way from adipose tissue to the epidermal epithelium. Whether that then has physiological consequences for the epididymis and the small RNAs that produce and stuff is the sort of million-dollar question and technically very difficult to get at.
One quick thing that I can add to this is that we did some luminometer studies in breast luminometer of all of male tracks from the HER2 mouse, and the epididymis does not oscillate, the testis does not oscillate, the liver oscillates beautifully. This is an arm hand, and the seminal vessels, very strong oscillators, but not the epididymis and not the testis as whole tissues.
It's fascinating. You set up where you have this transit through there, well, essentially there's different expressions, different. Have you ever done experiments where you just take photos of the epididymis and just do an asymptote to see how much of these are essential, these processes? There's a kind of experimentalist approach rather than a molecular one. I know you're not a biologist, but I'm curious as to how essential it is each of these. For example, cut out the middle part, cut out the end, the beginning, and then look at the function. Would the cells sort of readjust it?
Yeah, so I don't think anyone's done the kinds of experiments you're talking about. I think the transplantation would be very difficult, very small too. What people have done, and I never quite maintained this literature in RAM, but people have done a fair bit of either vasectomy or epididymal ligation in the corpus. And I think Pablo Visconti is one of the people who's done this. I believe if you ligate the corpus, the caput epithelium starts to express a more cauda-like gene expression program.
Yeah.
And I believe you can get some swimmers out of that. But again, those are all kind of like 50% certainty statements.
Per fatto specie abbiamo più persone di vedere queste opere, well, just I don't know much of the other. My question is, we study the exosome into the ejaculate by men, and so you think they mostly come from the epididymis or from other?
So I don't know, but I think generally speaking, people believe that for some reason they're called the ejaculasomes, for some reason get called seminosomes. Although I think that should more properly be used for the ones from the seminal vesicle. But I think, since in mouse, the seminal vesicle makes 90% fluid in the ejaculate, prostate like 8%, 6%, and then 4 to 5 for the epididymis. I don't know that the concentration of exosomes per microliter is the same across them, but if it were, then they'd be mostly seminosomes.
We found also in the single they also found some much lower than.
Yeah, but of course, very little fluid comes forward from the testis all the way to the ejaculate.
And then another quick question. We're planning to go back to see the sperm of the bee men in effect the birth weight, the obesity of the offspring. So I think that's a very efficient method. So the plan is to use smaller RNA. Do you think that's enough, or we should also that's the methyl or any other epigenetic marker?
Use small RNA, what to sort of to characterize the sperm of the obese men?
Yeah.
It's what I'd say is that probably 80% of my colleagues in the functional effects field are in the small RNA side of the hypothesis. So I guess that's and again, I sort of pointed out early on, that might just be because it's an easier one to test functionally, but it's also because of its role in RNAi and permutation. It's also sort of like the best hypothesis a priori, like the Bayesian prior is high. Low. Anyway, it's a probability. So yeah. Anyway, I think small RNAs are the best choice. They're also the cheapest choice because cytosine methylation is very expensive to look at if you want to do it carefully. And as I pointed out in one slide, nobody has ever measured sperm chromatin correctly, so you wouldn't even know what to do. I don't know what to do. So really small RNAs are way cheaper than cytosine methylation and also the best hypothesis at present.
Oliver, you showed the sperm dynamics in the male reproductive tract, but have you actually looked to see if there's further changes in the actual uterine, in the sperm that enter the uterus? Because there's a large filtration of sperm in all species virtually. So what you may get in the uterus may not be maybe completely different from what you get in the epididymis.
Right, and of course, all of the female reproductive fluids are filled with exosomes. The reason we haven't looked at that, for a variety of reasons. It's sort of hard to like scrape enough sperm out to clone stuff after mating and stuff. But the other reason is that because you can get these traits to go through IVF, it clearly can't be the female response to sperm, her changing the small RNAs, she delivers to sperm.
If you do it, if they're carried in vivo, it's an indication that they're carried.
But
I see.
if
So
you
you're-
do it in IVF, it doesn't mean that we may be doing something wrong in IVF that's not removing them all.
So what you're saying is that if we were to characterize the small RNA dynamics in sperm after introduction to the female reproductive tract, and all of the RNAs that we
Yeah.
look at in CAUDA are gone, then sperm RNAs are a very poor hypothesis.
Yeah. But if you're seeing, but I know that, natural mating, you see these effects, so I don't think you're wrong, but
Yeah, I
there
think,
may be some clean out of some sort via the female, in general, normally everything we do in IVF, the female takes care of a lot of the mistakes or a lot of barriers we choose to overcome, let's say.
yeah, so we are starting to pay attention to the female reproductive tract, in part because we're also interested in the question of the broader question of do the boy tubes talk to the girl tubes? And so we're quite interested in that question. And so as a result, we're sort of looking at gene expression and open up the rest of it after, with and without mating, with and without exosomes. Is that no answers yet?
I'm really glad I came today. We've learned a lot. So our next speaker. I don't have, everything I say doesn't have to be recorded. Got this. So our
No, you got it. 20 seconds. Sorry.